Good afternoon, everybody, and welcome to today's lesson on DNA mutations. Today, we're going to talk a little bit about the four major mutations that occur in DNA, and then look at some examples and see how that affects humans. Uh, humans and everything else. We're going to start just by defining these four different mutations. An addition mutation is exactly as it sounds. It is adding a new nucleotide. Right, so if we're starting with this sequence TACCG, an addition mutation would stick a new nucleotide somewhere in this list, right? Whether it would just put a new A over here or put a C here, right? It would add a new nucleotide where there isn't supposed to be that nucleotide. A deletion mutation is the exact opposite of an addition. It's removing a nucleotide from the sequence. Right, so if we're looking at this sequence, let's say I took out this T and now everything just kind of filled in, right? So instead of TATC, it would be TAC, right? Or I removed one of these A's, right? Those are deletion mutations. Now remember that DNA is read in groups of three, right? So they were going to read the TAC, CGT, ATC, AAG, and so on. So if you remove one of the, or if you remove or you add a new one, you shift the definition, the meaning of each and every amino acid trio down the line, right? Instead of this being read as ATC, now they're reading ACA, which is a different amino acid, right? A substitution is replacing a nucleotide. Replacing a nucleotide. Right, so instead of just adding or removing, it would be kind of both removing and adding. So it would take out this T and replace it with a C. Or it would take out this A and replace it with a G. Or it would take out, right? So it's going to take one of the nucleotides out and replace it with a different nucleotide. Which again is not going to shift every amino acid down the line, but it will, right? If this becomes a G, this is now a different amino acid than it was before. Right? And because we know that proteins are very dependent upon the order of their amino acids, changing even one amino acid can make a big difference in terms of what protein you get. Rearrangement is another one that's exactly like it sounds. It's shuffling the nucleotides. So whether that's taking TAT and rearranging it so it's ATT, or taking if taking TCA and making it CAT, it's just going to take and rearrange that clump, right? Whatever is targeted by the rearrangement mutation is just going to be rearranged. Sometimes it's as small as 2 just switching spots. Sometimes it's bigger, like 3 or 4 or 5 of them being rearranged. But either way, that's going to affect, again, the proteins that you make as it reads the DNA. And it's important to note that this is affecting the DNA, not the RNA, right? So this is affecting the permanent record of the cell, right? It's going to affect the DNA of the cell for the rest of time, right? Every new cell from that right, every new cell made from this cell with the mutation is going to have that mutation. All right, so looking at our 4 examples right here, addition added a C right here. Now we've got a new lineup of our nucleotides, a new reading of the DNA. Here for deletion, the G that was here is missing. So it got taken out, and now we're going to read, instead of CGT, it's going to say TAC, CTA, right, giving us a new lineup of all of these amino acids. They're all going to be shifted. Third, a substitution. This used to be a C, right? TAC, TAC. This was a C, now it's a T. It got replaced, substituted for another nucleotide. And the last one, rearrangement, is exactly the one that I pulled out before, right? This TCA is now CAT. They rearranged, right? Not they. The mutation caused the nucleotides to be rearranged, right? Giving us a new amino acid for this spot, or, right, let me see. 1, 2, 3. 1, 2, 3. Giving us 2 new amino acids because this affects ACA is not the same as ATC. All right. So, mutations. Mutations are dangerous and unpredictable because they change the amino acid sequence, like I said. If the amino acid sequence changes, the protein that you make is not going to be the protein that you intended to make, right? The fact that proteins are very dependent, right? Order determines structure, structure determines function. If you don't have all the amino acids in the right order, you are not making the correct protein. You can make something similar, but it will not be the one you intended to make, right? This is not great for us, mainly, right, in certain extreme terms. Things like sickle cell anemia is a mutation in your red blood cell shape. Instead of making red blood cells, the proteins that support the red blood cell and make it this shape are no longer making red blood cells that are like this little red bowl, right? Instead, you've got these sickle cells that can't hold the oxygen, right? Normally oxygen fits right into this thing and gets attached, right? Gets attached into the hemoglobin here, where in a sickle cell there's no little spot for the oxygen to attach to, right? And that's just a protein that is missing. Okay, so Atticus asks, are genetic mutations rare? It seems really easy for it to go wrong. It is really easy for it to go wrong, but mutations are actually a lot more common than you would expect. And the reasoning for that, right, is that my next slide? Yeah, so mutations are pretty common. They happen randomly. It's just a mistake, right? Imagine you had to copy a book word for word. You have to type up a book word for word. You're going to make a couple of mistakes. Most of the time, right, if you're typing and you're retyping a book and it says, that guy went, and you write, that guy went, it's pretty easy for the reader of that book to say, oh, this says that guy went. Or maybe it says this guy went, but determining in the context, it doesn't really change the meaning of the sentence that you're typing. When mutations happen, it's really easy for a mutation to happen in a cell that doesn't use the protein that got mutated. For example, if I have a skin cell that mutates, and now this skin cell, the gene for muscles gets messed up, that doesn't affect the skin cell, even though there's a mutation that happened there. Every skin cell that then follows that skin cell is going to have the genes for muscles mutated. But none of these are ever going to become muscles, so it doesn't really matter. Right? And that's true, right? Every cell in your body has all of the DNA that your body has. So it doesn't matter if the skin cell gets the gene for muscles messed up. It doesn't matter if a skin cell gets the gene that makes adrenaline messed up, because your skin cells don't make adrenaline. Right? Your skin cells don't become muscles. So even though it really messes things up for cells to get mutated, a lot of them are harmless, or they cause the cells to die instead of reproduce, or it's for something that doesn't really connect, right? Skin cells don't need the genes for muscles. Skin cells don't need the genes for adrenaline. Skin cells don't need the pigments that change your eye color. Right? All of those things are things that could be mutated, and it doesn't really matter. Right? In extreme cases, mutations are linked to cancers, right? It's not that all mutations are harmless, but it's not also that every mutation is something bad. Right? Mutations can cause cells to multiply out of control and become cancerous, or a cell could make a protein that it doesn't need to make, right? Make a protein that doesn't matter for it. Either of those things are possibilities. Right. Next time we meet, we're going to have a conversation about gene expression. So talking about how, like we said, the skin cell has the genes to make muscles. The skin cell has the genes that control your eye color. That's neither an E or a C. Right, has the genes to make adrenaline. Right, we're going to talk about how the cells themselves determine, well, do I need to care about this gene? Do I need to care about this gene? Do I need to care about this gene? Should I turn this on or should I turn this off? And we're going to talk about that next time I see you. Right. But the last topic that I want to talk about today is learned activities. Because a lot of people make the mistake of thinking that activities that you learn get coded into your DNA, and that's just not true. Right? The example I picked here, becoming ambidextrous does not change your genes. Right? Learning to write with both hands isn't going to mean that your children are then going to write with both hands. Learning to juggle, learning to ride a bike, these aren't things that get encoded into your DNA to get passed down. Also, mutations that occur in your body aren't a guarantee to be passed down to your children. Right? Is that here? Nope, not yet. Right? Anything that affects your genes that isn't in a gamete won't get passed down. If you have a mutation in your sex cells, that will get passed down. But anything else, any other mutation, is not passed down. Right? No matter what that mutation is, if it does not occur, or does not end up in a gamete, it will not be passed down to your children. Right? Only mutations in sex cells get passed down. Nothing that you learn will get passed down, right? Because those things don't get encoded into your DNA. Like I said, next time we're going to talk about gene expression and how that works in our bodies. But until next time, have a fantastic day. I'm going to transition us into tutoring and office hours. And you can come and ask any questions you want.